So today class is with continuation with the previous lecture I have delivered, and that is uploaded in my YouTube channel. The introduction to plant breeding. In the topic introduction to plant breeding, I have to be discussed the different types of the definition of the plant breeding given by the some scientists like Simmons as well as Vavilov, as well as some general definitions of the plant breeding, and I have also discussed the breeding objectives of the plant breeding and the nature of plant breeding. But today I have discussed here the other things that is also operated in the plant breeding, that is the activities of plant breeding. Activities of plant breeding means which type of work is conducted, which type of work is do in plant breeding, how to develop the variety, where to select the plants for hybridization, to making the hybrid. So all these things to creating, to making, to producing some important variety as well as hybrid, there are a procedure, a protocol follows, because we have to know that the definition of or nature of plant breeding. The plant breeding is the art, science and technology, and when we using this art, science and technology to improving the genetic character of the plant that is benefited to the farmer is called plant breeding. Or other definition of plant breeding is the genetic adjustment of plant for the social, cultural, economical and technological aspects for the environment. Ultimately, in plant breeding we have to change their DNA sequences in such a way that improve their trait in terms of yield, in terms of quality, in terms of disease resistant, in terms of insect resistant, in terms of early duration varieties, in terms of synchronous types of maturity, in terms of less amount of toxic content in the plant or as well as seeds. These are the objectives of the plant breeding. But in case of activities of plant breeding, the first activity is operated, that is the creation of genetic variation. Creation of genetic variation. Because when you willing to produce the hybrid as well as any type of variety in any crop, self-pollinated crop as well as cross-pollinated crop, first of all you have to be select the some genotypes, some parent. For the selection of parent for the making hybridization, for making hybrid, for making crossing, you have to be a population. Which type of population? The heterogeneous type of population. Those type of population where more amount of variability, more amount of variation is there, the differences are there, because we have to follow the selection procedure. So selection is effective in diverse type of population. Selection cannot effective in the similar type of population like the homogeneous type of population. Selection is always effective in heterozygous type of population, heterogeneous types of population. Heterogeneous types of population means different types of individuals in that population. Some plant have the 100 centimeter height, some plant have the 50 centimeter height, some plant have the 150 centimeter height. Means different types of their traits are different in different genotypes. The every genotype have the different type of their genetic trait. This type of population is called the heterogeneous type of population. Means dissimilar type of individuals are found in that population is called heterogeneous types of population. So the first activities of the plant breeding is to create or generate genetic variation in that population. Creation of genetic variation. So how do we create the genetic variation? We have to be generate or produce the variation in the plant with help of several types of methods like mutation, like polyploidy, like somaclonal variation, like hybridization, the several types of hybridization, intervarietal hybridization, interspecific hybridization, as well as intergeneric hybridization. Interspecific hybridization. Interspecific hybridization means hybridization between different species of the same genus is called interspecific. Inter means different or specific means species, crossing between different species of the same genus. I have given the example of the interspecific hybridization, that is the crossing between Oryza sativa with Oryza glabrema. Suppose you have to be crossing between Oryza sativa. Oryza sativa is the botanical name of rice, Indian rice, with Oryza glabrema, African rice. So genus is same, Oryza sativa, Oryza glabrema. Genus are same, but their species is different. One is sativa and other is the glabrema. So when this type of crossing, this type of hybridization is done, so this type of hybridization is called interspecific hybridization. Next is the intervarietal hybridization. Intervarietal hybridization means crossing between different varieties of the same species of the same genus is called intervarietal hybridization. For example, hybridization between Jaya. Jaya is a variety of rice. Hybridization between Jaya with IR64. So IR64 or Jaya both comes under the same species, that is the sativa. So a species is same, so of course their genus is also same, Oryza. So difference at which level? Difference is only for varietal level. One is the Jaya and other is the IR64. So this type of crossing is possible. So this comes under the intervarietal hybridization. And in plant breeding program, the several varieties are produced with help of intervarietal hybridization. Intervarietal hybridization is majorly used to making the variety, improved variety or hybrids. When we have to be produced or making the some disease resistant variety, so we going to the interspecific type of hybridization. The other type is the intergeneric hybridization. Intergeneric hybridization means crossing between different species or different genus of the same family is called intergeneric hybridization, where the genus is different. For example, triticale. Triticale is the first man-made cereals. Triticale is the first man-making cereals, crossing between wheat and rye. Triticale is produced from hybridization between wheat and rye. The scientific name of wheat is Triticum aestivum and the scientific name of rye is sickle cereal. So the sickle is the different genus and Triticum is the different genus. So this both plants comes under which type of hybridization? Both are comes under the intergeneric type of hybridization. So I hope that you have to be understand the three concepts, the three terms: the interspecific hybridization, intervarietal hybridization, as well as intergeneric hybridization. So with the help of hybridization, we have to be produce the variation in the population. Mutation also play a very important role to creating the variation in the population. Mutation is the sudden heritable changes in the character of an organism. Sudden heritable changes in the character. Heritable changes means those type of changes which changes carry forward generation after generation. Means if you have to be create the mutation in the first generation. So this changes, this mutant types is also observed in their progeny, because mutation is a heritable change, and heritable portions change means their DNA changes, and you have to be change the DNA sequence. Of course, these changes carry forward from parental generation to their progeny generation, their offspring generation. So mutation also play an important role to create the variation in the population. Same like the polyploidy also induce the variation. Domestication also induce the variation in the population. Selection also operates the variation in the population. So this is the meaning of the creation of genetic variation. Because selection is only possible when the heterogeneous type of plants are observed in that population. So this is the first activities of plant breeding, creation of genetic variation. Without differences in your population, you have not making any such type of variety or hybrid. You not getting the maximum amount of yield in your upcoming variety. You cannot imagine the maximum amount of heterosis possible in your hybrid. So this is the first activities of the plant breeding, creation of genetic variation. The next activities is selection. In those population where maximum amount of variation, all plants are different, so you have to be easily choose the best two plants. That plants have the different types of the character in the different forms. The variations at the different character level. The heights are different, the root lengths are different, the grain length are different, the number of branches are different. So for selecting the genotypes for the parent or selecting the parent, the both plants have difference possible at one character at least. At least one character is different for making the hybridization program, for making the hybrid. But you have to be more number of characters are different from both the parent. This is also very good to observing the heterosis in the progeny. So selection is also operated in the diverse type of population. The next activities of plant breeding is, after selecting the genotype, you have to be evaluate the selecting genotypes. Evaluation means comparing your selected genotypes from the diverse type of population, comparing with other high yielding varieties, other check varieties. Check varieties means check variety. I have also uploaded the videos in my YouTube channel. What is the check variety? You have to be kindly watch these videos on the check variety. Check variety means check varieties are most popular type of variety that is highly cultivated, highly preferred by the farmer. Check varieties is most growing variety by the farmer, most liking the variety by the farmer, most popular variety, high yielding variety. So this variety is called the check variety. This variety is used to comparing your new variety. So in the evaluate your selected genotype with comparing with the check variety, and after the evaluation, suppose you have to be selected the 10 varieties, 10 high yielding varieties. So out of the 10 variety, after comparing with check variety, you have to be got the only 5 variety or 5 plants, 5 genotypes or 2 genotypes. After comparing with check variety, the 2 genotypes are best after comparison. So the next activities after evaluation is the multiplication. Multiplication means those variety that compete with the check variety, the superior performance than the check variety. is to produce more number of seed. Multiplication means multiply their seeds because of the distribution to the farmer. All over the country, all over the farmer can easily find out that improved types of the variety, their seeds. So how to got the seeds of this improved variety? By help of multiplication. Multiplication means more number of seeds are produced of that compared genotypes. So this is the actual meaning of the multiplication. After the multiplication, you have to distribute the seeds to the farmer. So this is the activities in plant breeding. First, creation of genetic variation. Next, selection. After the selection, you have to evaluate. Then evaluation, multiply the seed, multiplication, and after that distribute the seed to the farmer. This is the main activities conducted in the plant breeding programs. I have also discussed some other things related to the plant breeding. A plant breeder should have the knowledge about some other disciplines like the botany, like the genetics, cytogenetics, plant pathology, entomology, bacteriology, plant biochemistry, plant physiology, as well as computer science. Because when the plant breeder making the some variety, during that time there are lots of knowledge are required. Lots of informations are required regarding development of the variety. Suppose when you have to be produce the disease resistant variety, so to making the disease resistant variety, so you should have knowledge about the plant pathology, because disease related pathogen related informations are available in the other subject, other discipline that comes under also the agriculture, the plant pathology. In case of plant pathology, in plant pathology we have to study the causal organism of the disease, which type of disease is occurring, how to produce the symptom. All these things are studied in the plant pathology. So a plant breeder must knowledge about all the sub discipline I have to be mentioned here. The knowledge of the plant pathology. So you have to be making the disease resistant variety. You have to be knowledge the plant pathology. You should have the plant pathologist. You should have knowledge about the entomology when you are making to the insect resistant variety. When you willing to produce the variety, that variety has not infected with any type of insect pest. So you should have knowledge about the entomology. So at a time, all the disciplines, all the information capturing in any plant breeder, this is not possible. So that reason we have to be consult, we have to be taking the information from the, we have to be included the some scientist of the entomologist, plant pathologist, plant physiologist, botanist. During the development of the variety, we want to include the some other scientist. We also include the some farmer. When we include the farmer during the development of the variety, this type of plant breeding is known as the participatory plant breeding. Participatory plant breeding, PPB. So what is the meaning of participatory plant breeding? Participatory plant breeding means participation. Whom participate in breeding program? The participation is for farmer. During the time, during the procedure of to making the variety, suppose you have to making the variety with the help of pedigree method or bulk breeding method. So during the selection of the genotype, at a time, during the time, the farmers are also included. Some selected farmers are, some progressive farmers are also selected during the development of the variety from the initial period. So this type of breeding is known as the participatory plant breeding. This helps the farmer to more understand, more knowledge about this variety, and the maximum chance to accepting this new variety by the farmer. After making the variety, this variety is maximum chance that is accepted by the farmer, because the initial time they are know about the everything. This variety is making according to the farmers, so the farmer is easily accepted. Otherwise, without included the farmer, your variety is very less chance to perform well in the farmer's field, or maximum chance the farmer ignore it, not accepted, not cultivated in our field, our area. So this is the actual meaning of the plant breeding. So in the both lecture I have to be dealing the very new information related to the plant breeding, because during the time of Green Revolution, the Green Revolution is achieved because of the some important reason, some important things, like we develop the high yielding variety. Green Revolution is possible due to the some important changes are taken place, and that changes is we have to be adopt some high yielding variety. The also other factor that is related to the Green Revolution is proper nutrient management, fertilizer. Intensive fertilizers are used during the time of Green Revolution, that is takes place in the 1966. Before the independence, the production of the agricultural food grain is very less. Before independence, the production of agricultural field crop is very low, only 50 million ton. But nowadays we are self-sufficient due to the high yielding variety. So how to making the high yielding variety? The high yielding variety is developed with the plant breeding. With plant breeding methods, the lots of technology, lots of techniques are incorporated in the plant breeding to making the some useful variety or hybrid. That hybrid, the performance, the yield of the hybrid or yield of the variety is more as compared to the traditional types of the variety, the land races types of the variety, the local variety. But the today variety is a very high yielding variety. All the population of the high yielding variety are looking same types of the progeny, the homogeneous types of the variety. So this is only possible with help of the plant breeding. And all the credits of the plant breeding and genetics goes to the Gregor Johann Mendel. Because of that reason, the Gregor Johann Mendel is known as the father of genetics as well as father of plant breeding. Because of, you have to be know that the name of Gregor Johann Mendel, he gave the law of dominance, law of segregation as well as law of independent assortment. So this is the information regarding the plant breeding, objectives of the plant breeding, nature of plant breeding, activities of plant breeding, as well as a plant breeding must knowledge about the some other discipline like plant pathology, entomology, bacteriology, plant physiology, botany, genetics, cytogenetics, and plant biochemistry, as well as plant biotechnology. So thank you.